Okay, good morning everyone. Okay, good morning all, good morning. Okay, so friends, in the last class, we were discussing about the eukaryotic DNA replication. If all of you quickly recall, friends, we have completed the prokaryotic DNA replication concept. And friends, we just started the replication in case of the eukaryotic organism. Let's continue. So I'll explain you for the period of 15 minutes, then I will solve all your doubts. So please continue. Friends, if you quickly recall, we understood that in case of a eukaryotic organism, the process of replication will occur little slowly. But friends, even the process of replication will occur little slowly, but the process of replication will occur most effectively. Is it? That is what we studied. And we read that there are multiple replicators also there. Then later we studied, friends, the type of enzymes that plays a key role is the polymerase alpha and polymerase delta. If you all quickly recall my words, polymerase alpha is the one which is actually acting like what? It is having the primase activity also, friends, and it is also having the polymerase activity also. But friends, we all people read very clearly this particular DNA polymerase alpha, even though it is having the polymerase activity, it cannot synthesize the daughter strand continuously. Is it? We read very clearly that it can synthesize the nucleotides up to 20 to 50 only because of its low processivity. So that's why what is happening, friends, the DNA polymerase alpha immediately can be replaced by what, friends? It can be replaced by the delta. Very simple point. So then I hope every student remember the process of initiation, how does it occur. And we also read very clearly about the regulation. So till here we have understood in the last class. I have told you very clearly, in case of a eukaryotes, the process of initiation occur in the two phases, correct na? One is the G1 phase, another one is the S phase. Friends, if you quickly recall, I have told you very clearly, in the G1 phase, pre-replication complex will form. Is it? Of course, yes. In the S phase, whatever the pre-replicative complex is formed, that pre-replicative complex will get activated. And we also read, my friends, the CDK level will be low in the G1 phase. And friends, we read that very clearly, the CDK level is high in case of the S phase. So this is what we have discussed in the last classes. That is still the process of initiation. Good morning all, please continue. Okay, so let me complete the elongation and termination. Later we will give the time to ask all your doubts. We'll clarify and we will move on to the transcription. Okay, so friends, let's start with the elongation process. What you have to understand the elongation, friends, we have read in case of a prokaryotic. Everyone please kindly look into the diagram. In case of a prokaryotic organism, polymerase 3 is the most powerful. Why polymerase 3 is the most powerful? What is the reason behind that? Because friends, polymerase 3 is associated with the beta clamp. That's why its processivity is more. It can easily synthesize the strands. But friends, here what I said, which is the most important one? Of course, friends, please kindly don't confuse here. Polymerase delta is the most important DNA polymerase. Of course, here we did not have any beta clamp, right? I want every student has to remember here the processivity can be increased just with the help of pCNA. Very simple point. Next one is what, friends? RFC, replication factor C. You know what is this particular RFC or replication factor C? Friends, it is actually acting like a clamp loader. So what do you mean by the word which is acting like a clamp loader, friends? In case of a prokaryotic organism, we did gamma complex, which is acting like a clamp loader, which can load the beta clamp onto the core enzyme of the DNA polymerase. Same here also. RFC is the clamp loader which can load pCNA onto the polymerase delta, which increase the processivity. Friends, everyone listen and look into the diagram very carefully. Understand. See, look into this. This is what, friends? DNA polymerase alpha. What is the speciality of a DNA polymerase alpha? Here, everyone look into the diagram, friends. Here you are able to see the green color. What is this? That is the primer. So who is adding that particular primer? DNA polymerase alpha. Friends, the moment DNA polymerase alpha add the primer, what is happening? DNA polymerase alpha itself will start elongating the nucleotides. So you can see the green color one is the primer, the red color segment is what, friends? Nucleotides. Listen carefully. How many nucleotides it can add, friends? It can add 20 to 50 nucleotide. That's all. After that, what is happening? Alpha can be replaced by what, friends? Delta. And we all commonly know, friends, in case of this particular leading strand, what we studied, there is only one primer can be added. That's all. Now that particular primer can be continuously elongated by alpha. Alpha can elongate up to 20 to 50 nucleotides. Then alpha replaced by delta. Now delta with the help of pCNA continuously start elongating this particular DNA in the leading strand. But friends, what is in the lagging strand? Everyone pay attention. There is one RNA primer and one Okazaki fragment. Once again, one primer, one Okazaki fragment. One primer, one Okazaki fragment. This is how the daughter strand synthesis will occur in the lagging direction, or it is occurring away from the replication fork. That's why what do you call it, friends? We can call it as discontinuous replication. That's why we are calling it as the lagging strand synthesis. I hope, friends, if every one of you very quickly recall what is the length of one Okazaki fragments, I said it to all of you. In case of a prokaryotic organism, the length of Okazaki fragment is 1000 to 2000. Is it? And they are present in thousands in number. But my friends, here the length of one Okazaki fragment will be 100 to 200. But friends, the point is very clear. How many number of Okazaki fragments are present? That is the question. Friends, millions of Okazaki fragments are present. Not one, not two, or not three. Millions of Okazaki fragments are present. Friends, pay attention. Like this, elongation will be continuously going on. So friends, once the elongation will get over, there should be, see, this is very important for the part B. Everyone pay attention. Who is going to remove the primers? Always compare with the prokaryotic organism. Friends, in case of a prokaryotic organism, who removed the particular primers, friends? Polymerase 1 is the enzyme which removed the primers, and polymerase 1 only replaced with the nucleotides. Finally, ligase enzyme will come and seal the nick. And I want every student has to remember the names very clearly. You know, right? In the leading strand there is only one primer, we all commonly know that. And in the lagging strand there are many primers are there. I want every one of you to understand one important point. All the primers, every single primer can be removed by an important enzyme. This is, we can call it as RNase H1. See friends, this is direct asker 2 marker question. All the primers removed by what? RNase H1. Friends, RNase H1 removes all the primers except the last one. Now the question is very clear, my friends. Who is going to remove this particular last primer? This particular last primer can be removed by an important enzyme. We called it as FEN1. You can ask me, sir, why RNase, listen to me carefully. You can ask me that why RNase H1 is removing all the primers. Why it is unable to remove only the last primer? I can use a word, because of low processivity, and listen my statements carefully. Suppose this is the primer, this is the strand. Now all of you listen, what is coming now? RNA polymerase. This is the this is the DNA polymerase. I'm sorry. The DNA polymerase enzyme will come fast. It has to stop here, right? See, this is the primer, friends. This is the primer. My right hand is the primer. My left hand is the DNA polymerase enzyme. DNA polymerase enzyme, actually when it comes to the primer, it has to stop. But you know what happened? The DNA polymerase enzyme will come very fast and it will hit the primer. Once it hit the primer, you know friends, this primer create like this. This primer create a flap. What it create? It create a flap like structure. So friends, because of that particular flap, see here, this is how a flap looks like. This is how the primer becomes now. Why? Because DNA polymerase enzyme coming very fast. It hit the primer. That's why primer create a flap like structure. So who is going to remove this particular flap like structure? Friends, this can be removed by an important and unique enzyme, which we call it as what, friends? FEN1, flap endonuclease 1. My friends, please kindly don't forget that. What do you call it as? Flap endonuclease 1. Once again, I am summarizing for all of you. Listen to me carefully. In case of leading strand or the lagging strand, all the primers can be removed by the RNase H1. Anyway, in case of a leading strand, how many primers are there, my friends? Just one primer, right? That one primer can be removed by RNase H1. Same way, in the lagging strand also, my friends, many primers are removed by what? RNase H1 only, except the last primer. Friends, that particular last primer can be removed by an enzyme called as FEN1. We can call flap endonuclease 1. Do not forget this particular statement. Now friends, this is the process of elongation. Now, everyone listen to me carefully. In case of a prokaryotic organism, I hope every one of you recalled, the process of termination is a very simple termination. Yes or no? If you quickly recall what we discussed, my friends, if this is the ORC, we discussed very clearly. One replication fork moves in the clockwise direction, another replication fork moves in the anti-clockwise direction. Both meet at one particular point. There the termination occurs. That is what we studied. But do you think eukaryotic replication process is really that much easy process, friends? No, not at all. Because I told you the reasons. Our genome is the most complex genome, and moreover, friends, our genome is what genome? Linear genome. So there are many complications will occurs during the termination of the eukaryotic organism. Everyone listen my statements very carefully, friends. See, whatever I am explaining in the diagram, the same thing was written here. Just focus the diagrams very clearly. Focus on the diagram. Look into this, friends. This is the origin, right? So from the origin, there are two forks are moving. I told you, right? In case of eukaryotic organism, ORR. In case of a prokaryotic organism, what did I say to all of you? The type of replication is the bidirectional replication. Is it? That is what we discussed, friends. Now all of you pay attention. One fork is moving in one direction, another fork is moving in the another direction. That we all commonly know that. My friends, after the successful replication, every student pay attention. After the successful replication, see here, finally we synthesized the two daughter strands. Every one of you pay attention. We synthesized the two daughter strand. Now, two daughter strand contain one primer. Two daughter strands contain one primer at the so you can see, right, there is a presence of one primer here at the 5' direction. Now student, just listen to me carefully. Now we have removed the primer. Who will come and remove the primer? As I told you, the last primers can be removed by FEN1. So FEN1, listen to me carefully, comes and remove the primer. There is no problem. After removing the primer, obviously we have to replace with the nucleotides. There is a gap created here, right? My friends, that particular gap we have to fill. We have to fill with what? We have to fill with the compulsory nucleotides. Now every student try to understand. Do you think DNA polymerases can replace the gap at 5' directions? Not at all. We read many times very clearly. If we want to replace the gap, what do we compulsory require? We compulsory require 3' hydroxyl group. Correct or not? When there is a presence of 3' hydroxyl group only, friends, then phosphodiester bond can be formed. But here you only tell me, here after removing the primers, what is there? 5' end is there. Now you only understand. No DNA polymerase is going to add the nucleotide at that particular 5' end. As a result, what is happening, friends? The length of the DNA becoming shortening every single time. So as a result, friends, the cell will, listen my statements carefully, as a result, cell will undergo the process of senescence, or cell will undergo the process of aging very fastly. Very fastly cell undergo the aging. Or there is a great chance the cell can convert it into cancerous very faster, because cell is not at all protected, my friends. Now what is the ultimate solution for this? Anyway, let me quickly repeat for all of you. What is the problem here? Our DNA is the linear DNA. So from the origin, two forks are moving, but one fork is in the clockwise or anti-clockwise. Now see here, when two daughter strands are synthesized, at the end, one primer is left. Where one primer is left? One primer is left at the 5' direction. Now, once we remove this particular primers, what happened here, my friends? All of you look into this. There is a creation of the gap. Correct na? Whenever there is a creation of the gap, DNA polymerases has to replace this gap with the help of nucleotides. But the question is very clear. If they want to replace with the nucleotide, there should be a presence of 3' hydroxyl group. But friends, there will be no 3' hydroxyl group. There is a 5' phosphate. It's very difficult to add the nucleotides. Polymerases are unable to add the nucleotide. As a result, friends, there will be a huge gap created of every cycle, which leads to allow the cell to undergo the senescence, which leads to allow the cell to become older very fast. Even it leads to the cell to enter into the cancerous condition. My friends, now what is the ultimate solution here? Every student, please pay attention. The ultimate solution for this is the telomere. Do not confuse, friends. See, this is the DNA, right? All of you understand the end structures of the DNA. Everyone friends, the end structures of the DNA, what do you call friends? So we will be calling it as the telomere. And I want every student has to remember 2 points about the telomere. What are the 2 points about the telomere that you have to remember? The first point friends, telomeres will increase the length of the DNA because it is adding at the terminal positions. By that friends, it can easily increase the end. Second one, it will protect the DNA from the degradation. What friends? It is protecting the DNA from the degradation by several vulnerable enzymes. So these two are the two major functions. I can tell you in one line. In one line I can tell you, if they ask you a part B question, what is the question? Which of the following enzyme is involved in maintaining the integrity of the DNA telomere? Once again listen, which of the enzymes are involved in the maintenance of the integrity of the DNA? All the students please remember friends, it's made up of telomere only. what I am saying, telomere can increase the length of the DNA, telomere can protect the DNA also. So friends, what is this particular telomere sequence is rich of? So mostly 99% the telomere is rich of T and G. Telomere is rich of what friends? T and G. Please kindly don't forget. And if you look into the structure of this particular telomerase enzyme, all of you listen friends, if you look into the structure of a telomerase enzyme consists of two components. What are the two components that the telomerase enzyme consists of? Friends, telomerase enzyme consists of telomerase RNA. At the same time friends, telomerase RNA it consists of, and it also consists of another important enzyme. That is, what do you call friends? Reverse transcriptase enzyme. It consists of telomerase RNA, it consists of telomerase reverse transcriptase. You all commonly know, right friends? Telomerase RNA function is what? Reverse transcriptase function is what? It will synthesize the DNA by using RNA as their template. Very simple point it is. It is able to synthesize the DNA using the RNA as the template. Now everyone please listen and look into the diagram very carefully. I'll show you the animation. Correct means correct. Thymine and guanine. Right. So that is what now I am saying. T and G. Look into this. See, this is the problem. See friends, this is our problem actually. One second, let me draw. What is the problem now? So this is the 3' and 5'. 5' got shortened, right? And 5' to 3' will be the normal. There is no problem. Friends, this is the exact problem. Now how this particular problem is getting repaired? All of you listen, don't confuse. What did I say to all of you? What does the telomerase enzyme is actually made up of? Telomerase enzyme is made up of telomere, then followed by telomere RNA, then followed by reverse transcriptase enzyme. Now friends, everyone listen, I'll show you the animation, you will get an idea. What this particular telomerase enzyme will do, you know, it has to replace the gap at the 5' end. But you know what it will do first friends? It will increase. What it will increase? It will increase this particular 3' end. All of you listen to me carefully. What does it will increase friends? It will increase 3' end first. Now you can ask me, sir, the problem is occurring at the 5' direction. Why the telomerase enzyme increasing the 3' direction? What is the reason? The reason behind that, whatever the strand it got increased, this increased part can act like what, my friends? Template. Very simple. So the extended part acting as a template. And what did I said to all of you? The telomerase having what? Telomerase RNA. Now friends, by using that particular telomerase RNA, reverse transcriptase enzyme will start synthesizing the nucleotide. The telomerase RNA, by using telomerase RNA, reverse transcriptase enzyme starts synthesizing the DNA. So I will directly show all of you in the animation itself. Just please look into this. I will explain here itself little elaboratively. Just a minute, you'll get a good idea here itself. Okay, whomever having the doubts, please look into this.
My job is to make college easier because students have a lot on them.
Okay, friends, look into this. This is the telomerase. Okay, and yeah, Reens, please look into your doubt. The telomerase, all see the arrow what I am showing, friends. The telomerase always having the rich sequence of T and G. I want every student has to understand one important word, friends. The telomerase enzyme, listen friends, every one of you, the telomerase enzyme always associated with the DNA in such a way that it will always form a overhang. Listen my friends carefully, whatever the arrow I am showing here, till here only our DNA is present actually. Till here only our DNA is present. So friends, just imagine this is the DNA. This is only the spectacles are DNA. Imagine my left hand is the telomerase enzyme. You know, telomerase enzyme associated with the DNA in such a way that it can form a overhang like this. It can form a extra overhang part. Always remember that particular point. Now listen. This sequence will be calling it as the telomere repeat sequence. The telomere repeat sequence are first identified in the tetrahymena actually. Okay. Okay. So it is always forming what, friends? Overhang. Now I want every student has to understand the structure. This is the repeated unit. And friends, what is our problem? Every student remember, the problem is this. At the 5' end, there will be a gap. Here also there is a gap. Here also there is a gap. Now we have to replace this particular gap with the DNA segments. Now for that, who is coming into the picture, friends? All of you please look into the diagram carefully. Telomerase enzyme coming. What does the telomerase enzyme consist of? Telomerase enzyme consists of telomerase RNA. At the same time, it also contains reverse transcriptase enzyme. Now, please kindly look into the arrangement, how it will exactly associated with the DNA molecule. My friends, everyone, please kindly look into the diagram. See, it is associated with the DNA in such a way that, see here, my friends, you only see, till G only the DNA is there, right? Till the G, that's all DNA. But see here, it form a overhanging structure. It always associated like that only. Now what it is doing, friends, instead of repairing the 5' end, what it is doing, what I said, it is going to extend the 3' end region. What it is doing, you can see in the picture, it will extend this particular 3' end. Why it is extending this particular 3' end, friends? Whatever the 3' end that it will extend, that can act as a template. Now see here, there is a complete extension. Now see here. Now it can, using this particular RNA as the template, it will start synthesizing the new strand. Very simple. But my friends, it has repaired the gap. But what is the new problem, my friends? The new problem here is it always form an overhang. Always form an overhang. That's why telomerase plays an actual key role in the process of cell aging and in the process of cancer. So friends, that is about telomerase or the termination in case of a eukaryotes. Friends, now tell me, is that clear for everyone? Yes or no? Come on friends, be active. Clear everyone? Reens, you understand now? Come on, be active. Clear everyone? Quickly give me the reply. Be active. Good, perfect. I hope everyone is active. Oh, great. That's good. I appreciate every one of you waking up early and listening the classes. I really appreciate. Okay, so friends, now let's start the next process, that is the transcription. My friends, yes, Saima, it contain the transcribal part. Yes, it do contain. Yes. Good. So friends, it contained here. Now finally we have finished the process of replication in prokaryotes and eukaryotes. I can tell all of you friends, we just started lesson. Okay, out of 100% of the lesson, we just finished 25%, I can say. Just that's all we finished. We finished very basic. I told you na, when listen my statements, all of you very clearly friends. When compared to the process of replication, transcription is a complex process. When compared to the transcription, translation is the more complex process. When compared to the translation, my friends, gene regulation is the little more complex process. Right now what we studied is just the process of replication. That's all. Now, as I already told you, if you want to understand the molecular biology in a crystal clear way, it is very important to connect all together. Once replication, transcription, translation over, automatically you feel the chapter is very easy. Now let's talk about, friends, the process of transcription, which is the most difficult process when compared to the replication. So I will explain everything elaborately, just pay attention. And we read, right, our ultimate goal of the DNA is what, my friends? Ultimate goal of the DNA is synthesizing the protein. But we all know very well our DNA never ever involved in the synthesis of protein directly. What it will do, friends, instead of involving in the synthesis of protein, the DNA is going to synthesize an intermediate molecule. My friends, what do you call this particular intermediate molecule? The intermediate molecule only we call it as the RNA. So all of you listen the definition, my friends. DNA mediated RNA synthesis. What, friends? DNA mediated or DNA guided RNA synthesis, which we call it as transcription. Now here comes the unique statement. What is that particular unique statement? Now you can ask me a question. Sir, are we using the entire DNA molecule to synthesize the RNA? That is absolutely wrong statement, my friends. If we want to synthesize this particular DNA, my friends, RNA, we will just use a small part. That's all. We will just use a small segment or the small part of the DNA only. Now you can ask one more question. Sir, which part you can say the part or the gene which is involved in the synthesis of protein? So if I want to synthesize the RNA, I did not require the entire DNA molecule. Only a little part of the DNA or a small segment of the DNA only required. Do not forget that, friends. So how can I define? Transcription is the process by which the information in a strand of DNA, not the entire information, friends, a small amount of information. Which small amount? The information which is actually coding for the protein. Only that information, my friends, it is going to get copied into a new molecules of mRNA. Friends, listen, when compared to the replication, more information you have to remember in the transcription. When compared to the transcription, little more extra amount of information you have to remember in the translation. So pay utmost attention. So there are three type of mRNAs are there: mRNA, tRNA, rRNA. Sorry, there are three type of RNAs are there. You know, right, friends? We commonly know that if the process of protein synthesis wants to occur, it is compulsory we require the 3 RNAs undoubtedly. And I want every student has to remember an important point. Among all, which is the most stable RNA, which is the see, they can ask a question, already asked question, which is the most abundant RNA in the animal kingdom, or which is the most abundant RNA in the animal body. I want every one of you remember that rRNA is the most abundant RNA. See, friends, if you look into the lifespan, of tRNA, friends, tRNA can withstand up to seconds to minutes. That's all. It did not have a greater lifespan. Friends, if you look into the mRNA can withstand minute to hour. But my friends, if you look into the rRNA, it can withstand up to generations. 2 generations RNA can withstand, friends. That much highly stable molecule the rRNA is. So always keep it in your mind. The most abundant RNA in the animal kingdom is the rRNA only. Friends, what is the function of mRNA? mRNA is the one which is bringing the blueprint. What do you mean by the word bringing the blueprint? Bringing the information. What information? It is bringing the message. What message? The information or the message which is actually translated into protein. So since it is bringing the message, we call it this as mRNA. Very simple point. Now next one, friends, tRNA. What is the point you have to remember? tRNA we can call the adapter RNA. What is the ultimate function of the tRNA molecule, friends? Ultimate function of the tRNA molecule is bringing an amino acid, adapting an amino acid. So information is there, amino acid is also there. By using this particular information with the help of amino acid, peptide bond can be formed with the help of this particular rRNA. So the point every student has to remember, if the process of protein synthesis wants to occur, my friends, it's compulsory. We require mRNA, tRNA, rRNA compulsory. You should not think that only if one RNA is present, protein synthesis can occur. No. 3 RNAs are compulsory in synthesis of this particular proteins. Friends, now actually before moving into the process of transcription, first we have to understand all the differences between the replication and transcription. Friends, most importantly, we have to study the structure of RNA polymerase enzyme. We should study most importantly the type of promoters. Once we study all these, the process of transcription will become extremely easy. Now, what are the fundamental differences between the DNA replication and transcription? See friends, very simple differences I will tell. I want you people to remember. We are commonly know that replication always occur in the S phase of the cell cycle, and transcription, that is synthesis of the RNAs, they will be taking place in the G1 phase and G2 phase. We all commonly know, right friends, G1 phase is the preparatory phase for what? G1 phase is the preparatory phase for the S phase. G2 phase is the preparatory phase for the M phase. So whatever the RNA that we are synthesizing, obviously we will synthesize in this particular Z1 and Z2 phases only. My friends, we all people read very clearly. See, this is one parent strand, this is another parent strand. If our DNA polymerase enzyme wants to synthesize the daughter strand, what it compulsory require, my friends? It compulsory require the primer. Correct na? So that's why, what is the statement I can tell? It requires the RNA primer for replication to start. But my friends, if you look into this particular transcription, you know, right, RNA polymerase is the more versatile enzyme, friends. It does not require a primer at all. Always remember that particular point. It does not require primer. And you only tell me, in case of replication, what happened? Two parent strands are separated and the two daughter strands are synthesized. Yes or no? What is the statement I can conclude here? I can conclude a statement that, friends, entire genome is copied. Yes or no? Of course, yes. We are going to copy the entire genome because the entire DNA is getting replicated. But you only tell me, I have already shown you in the diagram. Suppose this is the DNA. What we study, friends, only a little part of the DNA giving rise to the RNA. That's why what I can say, friends, only certain genes are copied. What, friends? Only certain genes are copied. Do not forget the statement at all. And friends, if you ask me very clearly, sir, which process is the most accurate process? Does the DNA replication is the more accurate, or the process of transcription is the more accurate? I can tell all of you, friends, the process of DNA replication is the more accurate process because of the enzyme DNA polymerase enzyme. DNA polymerase enzyme, friends, always try to maintain the greater accuracy, right? And what is this particular transcription, friends? Transcription is also an accurate process. I am not saying that transcription is not an accurate process. It is accurate process, but when compared to the transcription, DNA replication is more accurate, conserving the genome for further generations. And what is the ultimate goal of the RNA? Synthesizing copies of RNA of genes individually. Because we know, right, one gene, one protein, one gene, one protein. Very simple. So when the DNA replication occur, what we will get? We will get two daughter strands. And friends, when the transcription occur, we will get mRNA, rRNA as well as the tRNA. Very simple fundamental differences. Now friends, from this particular slide onwards, I want every one of you to understand very elaboratively. Okay, try to understand the depth of the concept. Friends, don't get confused. The site, all of you friends understand, the site where the process of transcription will start, the site at which the transcription will start, that particular site we called it as, friends, initiation site. Now every student has to answer my question, who is involved in the synthesis of RNA, friends? Of course, friends, RNA can be synthesized by versatile enzyme Rna Polymerase Enzyme. Friends, trust me, if you don't understand the prokaryotic transcription, it is extremely difficult to understand the eukaryotic transcription. That's why I encourage all of you to understand this very clearly first. Yeah, this is what site I am saying, initiation site. So friends, you know very well, RNA polymerase is the enzyme which will start the process of transcription. Here, what is the point I want you people to remember? RNA polymerase enzyme never ever associated with the initiation site directly. No, that is not possible. You should not think that directly RNA polymerase enzyme will come and associated with the initiation site and it start the process of transcription. No. First, friends, RNA polymerase attaches to the DNA at a particular site. What is that particular site? We can call it as a promoter. Friends, don't think that promoter is something else. Promoter is what, friends? Promoter is the a group of nucleotide, a sequence of nucleotide. That's all. What is the ultimate function of a promoter? Ultimate function of a promoter is that a promoter is the site where the RNA polymerase attaches. All of you understand, friends? First, RNA polymerase attaches here to the promoter. From here, RNA polymerase starts moving towards where, friends? RNA polymerase is moving towards the initiation site. It will come to the initiation site. Then only it start the process of transcription. Otherwise, it don't start the process of transcription. Now, friends, how many promoters are present in the prokaryotic organism? But if you look into the eukaryotic organism, especially humans, friends, we are having multiple promoters. Many promoters are there. But prokaryotic organisms are very simple promoters, friends. Then what is the statement you have to remember? It contains just two promoters. That's all. Only two promoters. One promoter is present at -10 nucleotides. Another promoter located, my friends, -35. What is this -10 and -35, friends? All of you pay attention. This is the initiation site, correct na? Yes, this is the initiation site where the transcription will start, where the promoters are present upstream. Which stream? Upstream. Where are left-handed side or the upstream promoters are present. So from the initiation site, if you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, at -10 nucleotide, one promoter is present. I have defined the promoter. What is the definition of the promoter? Promoter is the group of nucleotides, sequence of nucleotide. That's all, where RNA polymerase will come and associated. What are the sequences are present here? TA. That's why we can call it Tata box. And based on the name of the discoverer, my friends, we will be calling this as the Pribnow box also. Now, friends, once again, from the starting point, -35 nucleotide region also, there is another promoter is present, TTGACA. I am requesting all of you, friends, remember the sequences also. It's not difficult, right? TATA, TTGACA. Just remember this. And friends, all of you listen. 90% in the bacteria, only these two promoters are present. 90%. One promoter at -10, one promoter at the -35. Friends, two Indian scientists named as Richard and Gauss. What the Richard and Gauss identified, in case of a certain bacteria, not all, in case of a certain bacteria, even certain strains of E. coli only, there will be another promoter is located between -40 to -60, which is rich of A and T, where -40 to the -60. My friends, this particular -40 to -60 promoter not present in all the bacterial strain, in extremely particular strains only. Now let's talk. Friends, what is the ultimate goal of the promoter? I have told you very clearly. Promoter is the site where the RNA polymerase will come. RNA polymerase enzyme will come to the initiation site from the promoter. Then only, my friends, it is going to start the process of transcription. We all people commonly know that. Now let's talk about it. What is the most important enzyme that is involved here? the most important enzyme involved in the process of transcription is the RNA polymerase enzyme. Friends, I hope all of you remember, right? In case of a DNA polymerase in prokaryotes, what is the most important polymerase? DNA polymerase 3. I hope all of you remembered how many subunits are there in the DNA polymerase enzyme. 10 subunits are there. Correct na? Friends, now let's look into the RNA polymerase. Now, first point all the student has to remember, listen my friends everyone, in case of a eukaryotic organism, there are 3 polymerases are there, RNA polymerase 1, 2, 3. But friends, in case of a prokaryotic organism, there is only one enzyme. That's all. Only one polymerase enzyme will do all the process. Same. Friends, this particular RNA polymerase enzyme divided into different subunits. All of you listen to me. We can call it as the core enzyme. When the core enzyme, friends listen to me carefully, when it is associated with the sigma factor, when it is associated with the sigma factor, these two together, what do you call my friends? We will be calling this as holo enzyme. What do you call my friends? Holo enzyme. Holo, complete enzyme. So please listen, what does this particular core enzyme is made up of? Core enzyme is made up of 5 subunits. My friends, this is very important for the exam. You have to remember how many subunits are there, compulsory. So it contains 5 subunits: alpha, alpha 2, beta, beta prime and omega. Total how many subunits are there, my friends? Total 5 subunits are there. So when this 5 subunits of core enzyme connected to the sigma factor, then only we can call the holo enzyme. I want every student has to understand one very important point. If they ask you a question, always the statements will be confused. Even though you are having a crystal clear concept in your mind, the statements always confuse us in our CSIR exam. Now this statement is like this: sigma factor is an important protein is required throughout the transcription process. Once again listen, sigma factor is an important protein required throughout the process of transcription, which is absolutely wrong statement. You should not think that sigma factor is required all the time. No friends, sigma factor is just responsible for starting of the transcription up to initiation. I can say up to half of the initiation, not even in the complete initiation also. Because friends, everyone try to understand what I am saying here. Look into the diagram. This is the DNA, right? DNA is a very huge molecule, very huge molecule. On the huge molecule of DNA, there are 2 promoters are there. -10 and -35, 2 promoters are there. Now, this is the RNA polymerase enzyme. Now, you only tell me, how does the RNA polymerase enzyme comes to know? How does RNA polymerase enzyme recognizes where are the promoters on this particular double-stranded DNA? Because DNA molecule is very big. Promoters are present somewhere. But how does RNA polymerase recognize very accurately? Of course, my friends, with the help of this particular sigma factor only. Do not forget the statement. So, what is the statement I want to conclude here? I want to conclude a statement that sigma factor is only responsible for guiding the RNA polymerase. Why it is guiding the RNA polymerase? Just to recognize the promoter. That's all. It did not have an active role in the initiation, elongation and termination. It is only required for guiding the polymerase to recognize the promoter. That's all. Now friends, suppose example, this is the RNA polymerase enzyme. If you do the cross section of the RNA polymerase enzyme, friends, we will get different channels in the RNA polymerase. What are the different channels that we can observe? I want every student has to pay attention. You can see right here there is a channel called as friends downstream DNA channel. What do you mean by the word downstream DNA channel? Through which entire double-stranded DNA can enter. But friends, we read very clearly, RNA is how many stranded molecule? RNA is only one stranded. So we are having two strands in the DNA. Out of two strands, we will take how many strands? We will take only one strand, especially the strand which is in the 3' to 5'. So that is what do you call, friends? Whatever the strand that we are using for the RNA synthesis, we will be calling it as what strand? Template strand channel. Yes or no? Of course, yes. We will be calling it as the template strand channel. Now friends, you know right, how does the RNA comes out? This is the direction how the RNA is coming out. My friends, we will be calling this as RNA exit channel. What do you call, friends? RNA exit channel. We all know right, RNA actually comes out in the 5' to 3' direction. Always remember. Friends, in the replication we read very clearly how we can add the nucleotides, friends. We will always add the nucleotides at the 3' hydroxyl group only. We all people know that. So friends, this is what do you call, friends? NTP uptake channel. What do you mean by that, friends? Nucleotide triphosphate uptake channel, through which nucleotides are getting added. Very simple point it is. Okay, so friends, right now what we studied, we understand differences between replication transcription, we understand what are the enzymes required, we understand the fundamental differences also, and most importantly we understand promoter. Now everything is ready. Now we are going to see the process very clearly. The process of initiation, the process of elongation, and my friends, the process of termination. Always listen to me very carefully. Don't confuse. This is the slide where we are discussing the overview. In the next slides, we will explain one by one step very briefly and elaboratively. Just let me explain you the overview first. Friends, double-stranded DNA, how many strands we are using, friends? We are using just one strand. So which enzyme will come and associated? RNA polymerase enzyme. Everyone pay attention, friends. RNA polymerase enzyme want to associate it, always what does it require? Sigma factor. Friends, once the RNA polymerase enzyme recognize the promoter, you know right, we have to separate the double-stranded DNA. There is no doubt about it. When we want to separate the double-stranded DNA, what is the enzyme we generally use, my friends? Helicase enzyme we will use. We all know very well. But do you think RNA polymerase enzyme require the helicase? Tarun, I think you did not watch the animation very correctly, okay? Where you have seen that the telomere is associated with the 5' end. You did not watch the animation correctly, Tarun. Telomerase attaches to the 3' end. It is never attaches to the 5' end. Okay, you have seen wrongly. It will extend first 3' end. Later here the copy can be synthesized. So I am requesting you watch the animation once again in a clear manner. Okay, yeah. So once double-stranded DNA get opened, how it can open, friends? The RNA polymerase enzyme always having intrinsic helicase activity. What do you mean by the word intrinsic helicase activity? On its own, it can open the double-stranded DNA. It does not require the external helicase enzyme. Now, two strands are separated. I'm just telling you the overview. Now, one strand can be utilized as the template strand. Now, see, my friends, every one of you look into this. Sigma factor is dissociated. Actually, friends, in this particular diagram, they are showing that sigma factor is present throughout the initiation. No, I don't say that. Sigma factor won't be present throughout the initiation also, friends. When the initiation starts, sigma factor will be dissociated immediately. Now we know, right? We read many times, friends, in case of a eukaryotes, the process of transcription and the process of translation are coupled. What do you mean by that? Friends, what is coming out, friends? RNA is the one which is coming out. Now, friends, see here, RNA associated with what, friends? Immediately with ribosomes. Small ribosome and large ribosomal subunit will come and associate. They will start synthesizing the protein. So RNA is synthesizing. Simultaneously, protein also synthesizing. That's why transcription and translation are highly coupled. Then, friends, what is the another process we will be calling it as, friends? Termination. So when the process of termination will occur, there are different type of terminations are there. RNA, sometimes it come on its own, sometimes it cannot come its on own. So that's why whenever it is not coming out on its own, it is going to use an important protein called as a rho factor. That's why the termination can be divided into rho dependent termination, rho independent termination. This is just an overview of the 3 steps. Okay. So in the next class, friends, we will read the 3 steps very elaboratively from the starting. Okay. I hope it's clear for every one of you. So friends, from tomorrow onwards, the class will be up to 5:30 to 7 a.m. Okay. So today I am having one class at 7 a.m. in the Unacademy. So that's why I am ending the class now. So from tomorrow onwards, I'll schedule an Unacademy class at 7:30 a.m. So we will take the class from 5:30 to 7 a.m. Okay. So make sure that you have to attend 1.5 hour class continuously. By that I can finish the syllabuses very faster. Very, very fast we can do it. Okay. Clear everyone till this slide? I hope it's clear for all of you, friends. Great, 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 great. I hope it's clear for all. I am doing the ecology chapter in the Unacademy. If anyone wants to attend the ecology as a revision, you can attend it. The class is at 7 a.m. Class is at 7 a.m. Okay. I am really thanking every one of you waking up very early and attending the class. I really appreciate your responsibility. Thank you all. Thank you so much. I will see you tomorrow sharp 5:30 a.m.